Gas chromatography, or GC, is a powerful analytical technique used to separate and identify volatile components in a mixture. The separations in GC is based on two key factors. First, the differences in boiling points of the components, and second, how these components interact with the stationary phase inside the column. Now, GC operates on a two-phase system. Let me explain both phases. The mobile phase is an inert carrier gas. We use gases like helium, nitrogen, or hydrogen. The word inert is important here because the carrier gas must not react with our sample. It simply transports the vapor sample through the system. The stationary phase, on the other hand, is a liquid coating on the column wall or on a solid support material. This is where the actual separation occurs as the different components interact differently with the stationary phase. Think of the mobile phase as a river carrying goods. The carrier gas carries our sample. Meanwhile, the stationary phase acts like obstacles along the riverbank that slow down different boats by different amounts, causing them to separate. Next, let's understand how GC actually works. The separation process can be broken down into 5 sequential steps. Step 1. The sample is injected and immediately vaporized in the heated injector pot. The injector is heated to a temperature typically 50 degrees Celsius above the boiling point of the least volatile component to ensure complete vaporizations. Step 2. Once vaporized, the carrier gas, our mobile phase, transport the vaporized sample through the chromatographic column. Step 3. As the sample moves through the column, the components begin to separate. The separation is based on two factors: volatility, which is related to boiling point, and the interaction strength between each component and the stationary phase. Step 4. As each separated component exits the column, the detector identifies them as the elute. The detector produces an electrical signal proportional to the amount of compound passing through it. Finally, the signal is processed by a computer and displayed as a chromatogram, a graph showing peaks representing each separate component. Like dissolve like. This means that polar compounds interact more strongly with polar stationary phases, while nonpolar compounds interact more with nonpolar stationary phases. Most importantly, remember this key relationship. Compounds with lower boiling points elute faster than those with higher boiling points. This is because volatile compounds spend less time in the stationary phase and move more quickly through the column. Now, let's take a look at the specific objectives of these experiments. First, to determine the retention times of N-butanol and 2-propanol. Second, to identify the components present in a standard mixture based on their retention time. Third, to identify the component present in an unknown sample. Lastly, to determine the effect of temperature on retention time and resolution.
So let's start with the sampling injection procedure. So firstly, the shrink was rinsed before filling it with a sample. This is important to avoid the contamination. And then the shrink was gently tapped to remove air bubbles because air bubbles can affect the accuracy of the volume injected. And next, the shrink is held vertically with the needles facing up and the plunger was pushed to the required volume at eye level. So the excess sample was removed using a tissue. Now, let's look at how the gas chromatography system works as a whole. So first, we have the carrier gas such as helium or nitrogen, and this gas acts as the mobile phase and carries the sample through the system. And the sample is injected into the injection port where it is instantly vaporized due to high temperature. And after that, the vaporized sample enters the column, which is placed inside an oven, and inside the column, separation happens. Also, different compounds move at different speed depending on how strongly they interact with the column material. And once the compounds exit the column, so they will go to the detector, which detects each compound and sends signals to the computer. And finally, the data is displayed as a chromatogram, which shows peak representing different compounds. Next, let's talk about the main components of gas chromatography. So the first component is the carrier gas. It serves as the mobile phase and helps to transport the vaporized sample through the column. The gas must be chemically inert, meaning it does not react with the sample. And common carrier gases include helium, hydrogen, and nitrogen. So the second component is the flow or pressure regulator. So this part controls the pressure and flow rate of the carrier gas. A stable flow rate is very important because it can affect how fast the compounds move and how well they are separated. And the third component is the column, which is known as the heart of the gas chromatograph. So inside the column is the stationary phase. Different compounds interact with different phases, causing them to separate over time. Okay, moving on. So next, the oven. It houses the columns and control its temperature. Temperature control is very important because it affects how fast compounds travel through the column. And higher temperature usually makes compounds move faster. And next is the detector. The detector helps to identify and measure compounds as they exit the column, and it converts the presence of each compound into an electrical signal. And common detectors that usually used include the flame ionization detector (FID) and the thermal conductivity detector (TCD). Lastly, we have the display system such as a monitor or printer. This system helps to record the signal and display it as a chromatogram. And each peak represents a compound, and the height or area of the peak shows its concentration.
This chromatogram showed the pure compounds analyzed at 70 degrees Celsius. For 2-propanol, one peak was observed at a retention time of approximately 2.724 minutes. For N-butanol, one peak was observed at approximately 6.043 minutes. Each chromatogram shows a single peak, indicating the presence of one compound. I will explain how the peaks in the chromatograms were identified. Identification was carried out by comparing the retention time and elution order of peaks obtained at different column temperatures between the pure compound and the standard mixture. For the run at 70 degrees Celsius, two peaks were observed at 2.692 minutes and 5.903 minutes. The first peak was identified as 2-propanol, while the second peak was identified as N-butanol. Next, at 100 degrees Celsius, two peaks were again observed at 2.269 minutes and 3.294 minutes. The first peak remained as 2-propanol and the second peak remained as N-butanol. Lastly, at 140 degrees Celsius, the peaks were observed at 1.952 minutes and 2.225 minutes. Despite the peaks eluting closer together, the elution order remained the same, allowing consistent identification. Therefore, the identity of each peak was confirmed using the retention time matching and consistent elution order across all temperature. Based on Table 1, clear trends can be observed when comparing different column temperatures. At 70 degrees Celsius, what compound showed the longest retention times and the largest separation between peaks? At 100 degrees Celsius, retention time decreased and the separation between the two peaks became moderate. At 140 degrees Celsius, the retention time were the shortest and both peaks eluted very close to each other. Lastly, I will explain the effect of column temperature on chromatographic behavior. When column temperature increases, the retention time will decrease because the compound spend less time interacting with the stationary phase, which will also lead to a decrease in the peak resolution. Conversely, when column temperature decreases, the interaction with the stationary phase will increase, and this will result in longer retention time and improved peak separation. There is also a clear trade-off in this experiment. Lower column temperature gives a better separation but needs longer analysis time, while higher column temperature shortens the analysis time but compromises resolution. Importantly, despite changes in retention time, 2-propanol always eluted before N-butanol at all temperature, confirming consistent elution order. In conclusion, our experiment successfully demonstrated the fundamental principle of chromatography.
we were able to achieve a clear separation and identification of our two target compounds, which is 2-propanol and N-butanol. This success confirms that our initial parameters were well fitted for basic qualitative analysis. A key takeaway from this study was the critical relationship between column temperature and separation quality. We observed what is often called the statistical truth of gas chromatography analysis. While higher temperatures allow us to reduce analysis time and work faster, they often compromise the quality of the separation. Conversely, lower temperatures significantly improve our peak resolution, but they extend the analysis time. Essentially, we have shown that temperature and retention time share an inverse relationship that must be carefully balanced. Looking ahead, we can further refine this experiment in two ways. First, by utilizing gas chromatography mass spectrometry, this will allow us to use mass spectral analysis and library comparison to confirm molecular structures with absolute precision. Second, we could experiment with alternative stationary phases by exploring different column chemistries. We can enhance selectivity for specific compounds, potentially allowing us to maintain high resolution even at those faster, higher temperature. By understanding these variables, we can continue to optimize our chromatographic methods for both speed and accuracy.